Bismillahirrahmanirrahim. Quote of the day. Today is your opportunity to build the tomorrow you want. Assalamu alaikum. Today the topic of my presentation is types of vaccine under kind supervision of Professor Dr. Saira Afzal. Learning outline of my presentation is introduction about vaccine, different types of vaccine, example of each type of vaccine, recent updates, and at the end there is an MCQ session. By the end of this presentation, participants would be able to describe different types of vaccine along with examples. A vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. A vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed form of the microbe, toxin, or one of its surface protein. The agent stimulates the body's immune system to recognize the agent as a threat, destroy it, and to further recognize and destroy any of the microorganisms associated with that agent that it may encounter in the future. Vaccine can be prophylactic to prevent or ameliorate the effect of a future infection by a natural or wild pathogen, or may be a therapeutic to fight a disease that has already occurred, such as cancer. There are several types of vaccine: inactivated vaccine, toxoid vaccine, subunit, recombinant polysaccharide and conjugate vaccine, live attenuated vaccine, messenger RNA vaccine, and viral vector vaccines. Live attenuated vaccine contain whole bacteria or viruses which have been weakened so that they create a protective immune response but do not cause disease in healthy people. For most modern vaccine, this weakening is achieved through genetic modification of the pathogen, either as a naturally occurring phenomena or as a modification specifically introduced by a scientist. Live vaccine tend to create a strong and lasting immune response and include some of our best vaccine. However, live vaccine may not suitable for people whose immune system doesn't work, either due to drug treatment or underlying illness. This is because the weakened virus or bacteria could, in some cases, multiply too much and might cause disease in these people. For example, live attenuated vaccines are measles, mumps, rubella, or in the form of MMR, varicella, which is a vaccine for the chickenpox, influenza in the form of nasal spray, and rotavirus vaccine. These are the example of live attenuated vaccines. Now comes the inactivated type of vaccine. Vaccine of this type are created by inactivating a pathogen, typically using heat or chemicals such as formaldehyde or formalin. This destroys the pathogen's ability to replicate but keeps it intact so that the immune system can still recognize it. Inactivated is generally used rather than killed to refer to viral vaccine of this type, as viruses are generally not considered to be alive. Because killed or inactivated pathogens can't replicate at all, they can not revert to a more virulent form capable of causing a harm. However, they tend to provide a shorter length of protection than live vaccine and are more likely to require boosters to create long-term immunity. Example of inactivated or killed vaccine are polio in the form of injectables, poliovirus, or hepatitis A. Now there is a type of toxoid. Some bacterial diseases are not directly caused by a bacterium itself, but by a toxin produced by the bacterium. One example is tetanus, Its symptoms are not caused by the Clostridium tetani bacterium, but by a neurotoxin it produces. Immunization for this type of pathogens can be made by inactivating the toxin that causes disease symptom. This can be done by a treatment with a chemical such as formalin or by using heat or other methods. Immunization created using inactivated toxins are called toxide. Toxide can actually be considered killed or inactivated vaccine, but are sometimes given their own category to highlight the fact that they contain an inactivated toxin and not an inactivated form of bacteria. Example of toxide vaccines are diphtheria, tetanus, and part of DT acellular pertussis combined immunization. There are some advantages or disadvantages of toxide. Its product is devoid of live organism, implies greater safety, and multiple doses typically required, relatively expensive in manufacturing and cultivating of a pathogen for toxin production. Now comes the recombinant protein vaccine. Recombinant vaccine are made using bacterial or yeast cell to manufacture the vaccine. A small piece of DNA is taken from the virus or bacterium against which we want to protect and inserted into the manufacturing cell. For example, to make the hepatitis B vaccine, part of the DNA from the hepatitis B virus is inserted into the DNA of yeast cell. These yeast cells are then able to produce one of the surface proteins from the hepatitis B virus, and this is purified and used as the active ingredient in the vaccine. There are some advantages or disadvantages of recombinant vaccine. Those vectors that are not only safe but also easy to grow and store can be chosen. Antigen which do not elicit protective immunity or which elicit damaging response can be eliminated from the vaccine. For example, cholera toxin A can be safely removed from cholera toxin. And in the disadvantages, since the agent for the desired antigen must be located, cloned, and expressed efficiently in the new vector, the cost of the production is high. When engineered vaccine virus is used to vaccinate, care must be taken to spare immunodeficient. Subunit vaccine. Most of them are subunit vaccine which do not contain any whole bacteria or virus at all. Acellular means not containing any whole cell. Instead, these kind of vaccine contain polysaccharides or protein from the surface of bacteria or viruses. These polysaccharides or protein are the part that our immune system recognizes as a foreign agent, and they are referred to as antigens. Sorry. Even though the vaccine might only contain a few out of the thousand of protein in a bacterium, they are enough in themselves to trigger an immune response which can protect against the disease. Now comes the conjugate vaccine. Conjugate means connected or joined. When some bacteria to get protection from a vaccine, you need to train the immune system to respond to a polysaccharide rather than protein. But in the early days of polysaccharide vaccine, it was found that they did not work well in babies and young children. Researchers discovered that they work much better if the polysaccharide was attached to something else that create a strong immune response. In most conjugate vaccine, the polysaccharide is attached to diphtheria or tetanus toxide agent. The immune system recognizes these protein very easily, and this helps to generate a stronger immune response to the polysaccharide. One product information sheets, the diphtheria toxide is often called CRM197 carrier, which is a carrier protein because it is almost the same as diphtheria toxide, but not quite. Example of subunit or a conjugate vaccine is hepatitis B, influenza in the form of injection, Haemophilus influenza type B, pertussis, which is a part of DT, AP combined, and pneumococcal. Advantages of subunit vaccine: they can safely be given to immune suppressed people. They are less likely to induce side effects. And the disadvantages is antigens may not retain their native conformation, so that antibiotics produced against the subunit may not recognize the same protein or the pathogen surface. Isolated protein does not stimulate the immune system as well as a whole organism vaccine. Now comes the virus-like particles. Virus-like particles are a molecule that closely resemble viruses but are non-infectious because they are contain no viral genetic material. They can be naturally occurring or synthesized through the individual expression of viral structural protein, which can they then self-assemble into a virus-like structure. In some cases, the antigen in a virus-like protein vaccine are the viral structural protein themselves. Alternatively, the VLPs can be manufactured to present antigen from another pathogen on the surface or even multiple pathogens at once. As each virus-like protein has multiple copies of an antigen on its surface, it is more effective at stimulating an immune response than a single copy. For example, virus-like protein-based vaccine are currently used worldwide. Hepatitis B vaccine or a human papilloma virus vaccine, which is used against the cervical cancer. Outer membrane vesicles. Outer membrane vesicles are naturally produced by bacteria and are essentially a blob of the bacterial outer cell membrane. This contains many of the antigen found on the cell membrane but is a non-infectious particle. In the lab, these outer membrane vesicles can be harvested from bacteria to use as a vaccine. The outer membrane vaccine can also be modified so that toxic antigens are removed and antigens suitable for stimulating an immune response can be kept. Outer membrane vesicles also naturally act as adjuvants. For example, meningococcal B vaccine. This is a newer vaccine technology, so there are few licensed examples, and meningococcal B vaccine is one of them. Nucleic acid vaccine work in a different way to other vaccine in that they do not supply the protein antigen to the body. Instead, they provide the genetic instruction of the antigen to cells in the body, and in turn the cells produce the antigen, which stimulates an immune response. Nucleic acid vaccines are quick and easy to develop and provide significant promise for the development of vaccine in the future. Now comes the RNA vaccine. RNA vaccine use messenger RNA inside a lipid fat membrane. This fatty cover both protect the messenger RNA when it first enters the body and also help it to get inside cell by fusing with the cell membrane. Once the messenger RNA is inside the cell, machinery inside the cell translate into the antigen protein. This messenger RNA typically lasts a few days, but in that time, sufficient antigen is made to stimulate an immune response. It is then naturally broken down and removed by the body. RNA vaccines are not capable of combining with the human genetic code. There are two RNA vaccine authorized for emergency use in the UK at present. The Pfizer BioNTech and the Moderna COVID-19 vaccine are both RNA vaccine. Now comes the DNA vaccine. DNA is more stable than messenger RNA, so doesn't require the same initial protection. DNA vaccine are typically administered along with a technique called electroporation. This uses low-level electronic waves to allow the body's cells to take up the DNA vaccine. DNA must be translated to messenger RNA within the cell nucleus before it can subsequently be translated to protein antigen, which stimulate an immune response. There are currently no licensed DNA vaccine, but there are many in development. Viral vectored vaccine. As with nucleic acid vaccine, viral vector vaccine are a newer technology using harmless virus to deliver the genetic code of a target vaccine antigens to cell of the body so that they can produce protein antigens to stimulate an immune response. Viral vector vaccine are grown in cell lines and can be developed quickly and easily on a larger scale. Viral vector vaccine are significantly cheaper to produce in most cases compared to nucleic acid vaccine and many subunit vaccine. Replicating or a non-replicating, there are two categories in a viral vectors. Replicating viral vectors retain the ability to make new viral particle alongside delivering the vaccine antigen when used as a vaccine delivery platform. As with the live attenuated whole pathogen vaccine, this has the inherent advantage as a replicating virus that it can provide a continuous source of vaccine antigen over an extended period of time compared to a non-replicating vaccine, and so is likely to produce a stronger immune response. A single vaccine may be enough to give a protection. Replicating viral vectors are typically selected so that the virus themselves are harmless and are attenuated. So whilst they are infecting the host, they cannot cause disease. Despite this, as there is still viral replication going on, there is an increased chance of mild adverse event with these vaccines. A vaccine to prevent Ebola called Ervobo, used as a recombinant vesicular stomatitis virus. This vaccine was approved across Europe for use in 2019 and has been used in multiple Ebola outbreaks to protect over 90,000 people. The vaccine has primarily been used in ring vaccination, where the close contact of an infected person are vaccinated to prevent the virus from spreading. Now comes the non-replicating. Non-replicating viral vectors do not retain the ability to make new viral particles during the process of delivering the vaccine antigen to the cell. This is because key viral genes that enable the virus to replicate have been removed in the lab. This has the advantage that the vaccine cannot cause disease and adverse events associated with viral vector replications are reduced. However, vaccine antigen can only be produced as long as the initial vaccine remain in infected cell, most likely a few days. This means the immune response is generally weaker than the replicating viral vector, and booster doses are likely to be required. A viral vector vaccine developed to prevent Ebola was licensed for the use by the European Medicine Agency in July 2020. The Oxford AstraZeneca COVID-19 vaccine, which was approved for the emergency used by the MHRA in December 2020, and also uses a non-replicating viral vector called CHA-DOX1. Now it's time for the recent updates. New vaccine in development. Vaccination was first planned in Europe, and ever since, Europe has been at the heart of the global vaccine industry and playing a key role in the research and the development of the vaccines. There are many new vaccines currently being researched and developed within our industry, including use of viral vector vaccine, non-replicating vector vaccine, RNA vaccine, DNA plasmid technology, and immunotherapy. A number of vaccines in development target disease that are difficult to treat, such as HIV/AIDS and other viral diseases like cytomegalovirus, dengue, Ebola, and cancer. Alzheimer's disease, rheumatic disorders, bacterial diseases like Clostridium difficile and Chlamydia E. coli, or some parasitic diseases like malaria, hookworm infection, and leishmaniasis. In addition, several combination vaccines offer practical advantages that can boost vaccine uptake, thereby further improving public health. Vaccination against Ebola, one of the most significant leaps in vaccine development and collaboration in the last 30 years. Since Ebola was discovered in 1976 and there have been around 30 significant outbreaks in Africa, the 2014-16 epidemic alone killed more than 11,000 lives West Africa and had a devastating impact on families, health system, and economics. At the time, there was a little to stop it. There were no effective treatment or approved vaccine. But in 2018, Ebola reared its head again in the Democratic Republic of the Congo in the world's second largest outbreak, causing more than 33,000 cases and 2,000 deaths. To date, two different vaccines have been developed. In November 2019, MSD's single dose vaccine offering was approved in the Europe, and a few months later, in July 2020, Janssen two doses regime was approved in Europe for the treatment of Ebola. Now it's time for the multiple choice question. Live attenuated vaccine contain whole bacteria or virus which have been weakened so that they create a protective immune response, not causing disease in healthy people. Which of the following is not the example of a live attenuated vaccine? Tetanus vaccine, MMR vaccine, varicella vaccine, influenza vaccine, or a polio vaccine?
A.
A is the right answer, tetanus vaccine. A subunit vaccine contains a fragment of the pathogen and elicit an appropriate immune response. Subunit vaccine is all except a whole purified viruses, a purified part or a piece of the antigen, an expensive type of vaccine, hepatitis B vaccine, or MMR vaccine.
C.
A is the right answer, a whole purified virus. Which of the following type of vaccine do Johnson & Johnson and AstraZeneca belong to? Live attenuated, messenger RNA vaccine, viral vector, toxoid vaccine, or a subunit vaccine?
B.
B, messenger RNA is the right answer. Which of the following statement is incorrect about the live attenuated vaccine? It is prepared using whole weakened living bacteria or virus. It can generate a long-term immune response with the administration of a single dose. Measles, MMR, and oral polio vaccine are live attenuated vaccine. It is stable at normal room temperature. Varicella is example of live attenuated vaccine.
D.
D is the right answer. Yes, D is the right answer. It is stable at normal room temperature. It has been reported that thousands of women worldwide are affected by cervical cancer and equal number of men contract sexually transmitted disease annually. Human papilloma virus, the common microorganism that is associated with cervical, vaginal, or anal cancer, can be protected by the vaccine, which is a type of live attenuated, killed, toxoid, virus-like particle, or messenger RNA vaccine.
B.
D is the right answer, virus-like particle. Two example of this type, hepatitis B, uh, it can be treated, or human papilloma virus vaccine is the example of virus-like particle. All of the following vaccine are example of the recombinant type vaccine that are currently used in developed countries, except vaccine for hepatitis B, vaccine for human papilloma virus, vaccine against Haemophilus influenza B, or vaccine against hepatitis A.
D.
D.
D is the right answer, vaccine against hepatitis A. Killed vaccine are made by destroying the pathogen's ability to replicate but keeping it intact so that the immune system can recognize it. Select the incorrect statement regarding the inactivated type of vaccine. Prepared by using heat or radiation for the inactivation of toxin of the microbe. Most effective against bacterial pathogen but less on viral pathogen. Poliovirus and hepatitis A are common inactivated vaccine. Prepared by using heat or radiation for the inactivation of the microbe.
B.
A is the right answer, prepared by using heat or radiation for the inactivation of toxins of the microbe. Virus-like particles are a molecule that closely resemble virus but are non-infectious because they contain no viral genetic material, and it can be naturally occurring or synthesized through the individual expression of viral structure protein, which is the example of VLP vaccine, hepatitis A, hepatitis B, polio vaccine, MMR, or varicella vaccine.
B.
B is the right answer, hepatitis B vaccine. Before the invention of the polio vaccine in 1955, poliomyelitis was a serious viral infection. Thousands of children got infected and died around the world. Select the incorrect statement about the polio vaccine. It is only present in the form of killed or inactivated vaccine. Live virus vaccine against poliovirus can be given orally. The dose of vaccine must be given before children turn 6 years of age. It is present in two forms, oral polio vaccine and inactivated polio vaccine.
A.
A is the right answer. It is only present in the form of kill or inactivated vaccine. Outer membrane vesicle are naturally produced by the bacteria that contain antigen, although being a non-infectious. These can be harvested from the bacteria to use as vaccine. The only vaccine use this new technology is hepatitis E, COVID-19, herpes simplex virus, meningococcal B, or a polio virus. OMV.
B.
A D, meningococcal B, is the right answer. This is the key of my MCQs, references of my presentation, and the YouTube link of this presentation. Thank you.
Thank you so much, Dr. Naila. Will you please stop the recording?